diseases. Okay. And so I'd like to start, again and feel free to either, unmute yourself and ask a question or type in the chat. I should see your question in the chat. Realize that, infectious diseases are, they're rather prevalent in medicine. However, not every microbe, causes an infection or infectious disease. And so, there are, I don't even know the number, but millions of different types of microbes that are in the environment. And it's only select, it seems like a ton when you're studying them, but there's only a select number that are really pathologic for human beings. Okay. And so there is this kind of ongoing dynamic between the environment, the pathogens, and the host. And we're specifically talking about humans at this point in time. So, number one, you have to have, we're going to talk about some of these different concepts. You're learning epidemiology in your epidemiology class. And so that really studies it kind of studies the environmental aspects of the disease and how disease gets transmitted, etc. From a medicine standpoint, that's all important, but our focus is on how the disease is presenting in the host itself and how do we treat it and cure the disease. And from the agent's perspective, it's really about, okay, what's in this for me? And, because they rely on nutrients and, and metabolic processes from outside their environment, because the majority of these are single cell pathogens and microbes, and they have to get their nutrition and resources from other sites. So for them, it's just another meal, and a place to live. So, but there are factors that enable these hosts to infect, and there are environmental aspects to all of this. We talk about the external environment, the internal environment of the host. And so, lots of conditions can promote infection. So this is kind of the famous, kind of triangle of interaction between those three components. Starting with some terms and definitions, which you've probably already known. The host is really the living organism. Hi, buddy. The living organism that can supply the microbe or another with whatever the growth needs are. So that becomes the host, the source of all of the nutrition. We always look at this continuum of infection that often starts with colonization. Okay. And, colonization is on one end of the spectrum. Septic shock and death is on the other end of the spectrum. And so many infections can start with colonization. At which point does the scale tip in the middle or wherever, in that this colonization then becomes an infection, then becomes sepsis, then becomes septic shock. And so it's all a continuum. And if we have a healthy immune system and our immune cells are working properly, along with many other factors, we may either keep the pathogen at bay or keep it as colonization, where there's no harm to the host itself. It's only when there's harm and manifestations of disease that we call it an infection. Like, as you know, we have, normal flora in various parts of our body, and they coexist and they can actually be healthy for us. And so, you know, some of um, some of them can be, when we have commensalism, it's really one organism is sustaining the growth of another, but it doesn't mean that there's any detriment to the host itself. Parasitism, there's often a detriment. So a parasite will live off of another organism, often at that organism's expense. Mutualism, both benefit. So, a good example of mutualism is like some of the E. coli in our large bowel that produce vitamin K. Yes, it gets its food source and can, live out its lifespan and multiply, etc., in the large bowel, but it provides us with vitamin K, which then is absorbed through that gastrointestinal mucosa and utilized in the manufacture of many of our clotting factors. The virulence, it has to do with the disease causing potential, and that is an important factor in that continuum of infection. And then pathogens are typically organisms that are capable of causing disease. It doesn't mean that they're always going to cause disease, because we come in contact with pathogens all the time. But our immune system is able to, essentially, take their pathogenicity away, kill them, isolate them, and not allow them the opportunity. So again, that kind of assumes a, healthy immune system. So there's a lot that goes into, the development of infections. Looking at some of your common pathogens, and this is a chart taken from your text, the list is there, which we've gone over before, okay? And so this is just a good summary slide, and it does have some of the functional characteristics as well as how do we treat them, and it's general, and it gives you some examples of these different pathogens. Okay. If you compare some of the characteristics, realize that, prions, viruses, bacteria, all, your mycoplasmas, which are a specific type of bacteria, as well as, your spirochetes, your rickettsia, and your chlamydia species, they all are prokaryotes, and they have no defined nucleus other than prions. Prions a protein. It is not a living organism as we have defined an organism. And so none of these have a defined nucleus. When you get up to yeasts, which are unicellular, versus molds, which are multicellular, okay, these become eukaryotic. They have a defined nucleus, okay, as well as your protozoans and helminths. Most of these organisms, and I use organisms lightly when it comes to prions, are DNA based. However, viruses can be DNA or RNA, as you've already learned. And prions are proteins. They're irregularly folded proteins and with no known genetic material. And this just kind of goes about their size, and then, this talks about whether they're intracellular or extracellular, okay? And then do they have any motility? And so that you can see that some bacteria may be motile. Your spirochetes, by their structure, are motile. Clearly, your protozoans and your helminths are motile, okay, which gives them an added virulence factor. So we'll start with prions. I got a chat question. What is the intracellular, extracellular referring to? Okay, so the intracellular, extracellular is referring to where they are found. Okay, and how they where they're found in the environment or in even in the body. So prions are extracellular even in the body. Okay? They don't invade intracellularly. Viruses, it's obligatory that they become intracellular for reproductive purposes and growth and everything else. Bacteria can be either in the body. Your mycoplasmas, they can reproduce extracellularly. However, they can be found intracellularly in the body. They have a predilection for certain cells, which you'll learn as we go on. And so you can go through and take a look at these. Okay, prions themselves, okay, these are protein particles. They are self-propagating, okay? They're able to transmit infection, even though they're not considered technically an organism because they lack DNA, but they can propagate more prion proteins. Okay, and so some of the diseases are listed here, okay, that they cause. Essentially, these are very transmissible particles that cause a transmissible neurodegenerative disease. And regardless of which one you see here, they are neurodegenerative disease. Because they are unusual and they are resistant to proteases, okay, we have no treatments for these diseases. So essentially, they're all fatal. Now, the most recent pathology, what they're studying what they're theorizing occurs is we actually have other prion proteins that are on our cell membranes. Okay? And what happens is you get exposed, whether it's by eating contaminated, improperly cooked meat, or in the case of animals, whether it's, drinking from a contaminated water source. And these prion proteins get into the brain, okay? And you have these abnormal folding. Well, what they're saying is that there are prion proteins that are normal, okay, that are on the cell surface membranes. And when these abnormal ones get inside the brain, they come in contact with the normal ones on the cell membranes. And when they do, it transforms those prion proteins into the abnormal protein. And then essentially it gets released from the cell membrane, and that's how it self-propagates. The cell then will replace that normal prion protein with a new prion protein that's still normal, okay, but it allows this abnormally folded proteins now that are, multiplying to then bind again and extract these prime proteins from these normal proteins from the cell and transform them into these abnormal prion proteins. And as they collect inside the interstitial space in the tissues themselves, they will aggregate. And that's what this is showing in this middle diagram here, is they will actually then aggregate. And then much like in Alzheimer's disease, they will form these kind of plaques and tangles that then disrupt normal neural communication, which then will result in the degeneration of healthy neurons within the brain. Okay? And so cellular precursors in the actual cell membranes are what are being recruited by these abnormally folded proteins, which then leave the cell membrane, form aggregates within the tissues, okay, and between the cells, and keep recruiting more and more of these proteins, which build up these plaques and interfere with neural transmission, leading to the degeneration of neurons in the brain. That's your, transmissible neurodegenerative disease. Are there any questions on that? It's pretty fascinating. Okay, so that is the current thought process with prions. And because they're resistant to proteases, they don't have a cell membrane, they don't have DNA. Okay, so if you go back and look at all of our antimicrobials, they all have very specific targets. There are no ribosomes. There are no ribosome subunits. And so none of our antimicrobials are effective against these prime proteins right now. So 100% fatal. All right, viruses. Okay. We've talked ad nauseum about viruses, and we've had a lot of experience lately with viruses. And these are the smallest of all obligate intracellular pathogens, okay? And so because there are no organized cellular structures inside, okay, it requires the machinery, if you will, of a specific host cell to insert its DNA, take over the function of the cell to then reproduce more virions, right? They contain a protein coat, also known as a capsid. They have a nucleic acid core or genome, and it can be RNA or DNA. Okay, some will contain that envelope, like Hep B has an envelope. That's why you can develop antibody to the envelope protein. And the envelope itself is actually lipoprotein that is derived from the cell that it leaves. So as these viruses are reproduced, it's essentially like exocytosis, so that once the genome is formed, it kind of like buds outward and is covered by a piece or a coating of that cell membrane lipoprotein layer. Okay? And that becomes that envelope. Okay? And so your herpes viruses and paramyxoviruses are common viruses that do have this envelope protein, this lipoprotein envelope. And, and it continues these in that way. The host cell is not lysed necessarily. And so that host cell can continue to produce more viruses. Sometimes the host cell is destroyed. Right? And we see that with CD4 cells in HIV infection. Okay. And so how do we characterize our viruses? There's a lot of different ways to characterize them. They can be grouped as like a DNA virus or an RNA virus. You'll hear that often. Sometimes it's physical characteristics. Sometimes it's how they replicate. Sometimes you'll hear, oh, this is a respiratory virus. So mode of transmission, okay? And there are multiple respiratory viruses, okay? And they come from different, genuses, if you will. They come from, you can characterize viruses by the type of target tissue, like your hepatitis viruses. And again, you've got all these different A, B, C, D, E. Okay, so you've got all these different hepatitis viruses. But as you look at those viruses, they're very different. They're very different. Okay. So here's your basic structure, and with or without an envelope. So your basic virion, okay, with your DNA or RNA, okay? And then you have your envelope, which is lipoprotein that is essentially pinched off as it exits the cell. It's part of the host cell's outer cellular membrane, okay? And so with viral replication, okay, once that infects a host cell, okay, we're going to start with, let's see, we can start with the cell up here at the top at 12 o'clock. And you have attachment of the virus, which then has to somehow enter the cell. Sometimes that can be a key to treatment. If we can prevent it entering the cell, that can often be a key to treatment. Okay. And so you have a virus that enters a cell. Okay. It penetrates, gets into the cytoplasm. You get uncoating of the viral capsid or envelope. Okay. Which then allows the DNA or RNA to be freed up. So then it can utilize the host cell's structures to replicate, okay? And depending on which viral type it is, formulate the protein coat, okay? Mature these viruses, which then can lead to, in the case of enveloped viruses, bud out, pinch off, taking a piece of that lipoprotein membrane with it as it goes. Okay? Or in the case of other viruses that are non-enveloped, that they can lyse or rupture the host cell, freely releasing virions into the environment, whatever environment that is. Right? Somewhere along the line, there is the potential for latency, where the genome can lay latent within a host cell, embed into the genome. It can switch on oncogenes, which we've recently talked about, leading to a malignancy. And that can occur within years, decades, It just all depends. Or it can lay in latency until the opportune moment, as in the case of, varicella zoster, and then activate, pick up the life cycle, and then create disease again. So there are multiple options for some of these viruses in which direction they go. Okay. If you look specifically at Epstein-Barr virus, okay, and it has multiple pathways. It can lead to infectious mononucleosis, okay, which is kind of an acute infection with acute symptoms. It can lead to a chronic low-grade infection, and this is within the herpes virus family, and so it can become latent. We know that chronic fatigue syndrome is associated with infectious mono and the Epstein-Barr virus. We know that it can transform into malignancy, both pharyngeal carcinoma as well as Burkitt's lymphoma. Okay. And so you have the first exposure to saliva. The virus, whatever the means of transmission, drinking from a straw, glass, eating from a utensil, kissing, it is called the kissing disease. Okay. And that virus will get into the pharyngeal epithelium. Okay. And that can be it can then switch on genes to lead to nasopharyngeal carcinoma. You can have activation of the viral cycle. It can cause all of the signs and symptoms of acute infectious mononucleosis. It can also be incorporated into B cells, where you get activation of your B cells and change into transformation into Burkitt's lymphoma. Okay. And it can stimulate the atypical lymphocyte, and some of them can either kill more T cells or actually suppress your immunoglobulin response. So there's all kinds of different ways that this can occur. Is the process of viral entry into the cell what I was referring to when we were learning about lifegenia? Yes, it is how it enters into the cell. And yes, in that particular case, it's altering the DNA of those specific cells. And yes, it targets specific proteins that are, cell specific, tissue type specific. So that's why, like your hepatitis, A virus, it's not going to cause a respiratory infection because it doesn't have a predilection for the surface proteins of your alveolar cells. It's going to specifically search out its receptor site on a hepatocyte, if that makes any sense. Okay, awesome. All right, there's a ton of viruses out there. There's just a ton of viruses out there. I'm going to focus on some interesting ones. Okay. And so our influenza viruses, are quite fascinating, really. They come from the Orthomyxoviridae family, and there are many, many variants. Okay. Of specific interest are the H5N1 and the H1N1, and these are the H5N1 is the avian flu, and H1N1 is swine flu. Okay, and both of these one of the very, very interesting things about influenza, and especially these particular variants, is that they originated in animals. And for the longest time, there was no crossover into humans. I went to a really interesting lecture one time where they talked about this whole how it jumps species and it learns to jump species. And one of the places where, and this is all, this is not throwing stones or saying anything bad, but when you have humans in constant contact or in the same environment as other animals, and there's close physical contact constantly, you run the risk of it learning enough information to then jump species. And there's a whole lot of epidemiology behind all of this. But rice paddies in China tend to be one of those places where you will see, it tends to be swampy with the growth of the rice. You have humans out there, you have ducks and other, avian animals, mammals for the lack of a better term at this point. You have swine, pigs, you have dogs, you have, and all of this comes together in this great mixing pot, if you will. And all of a sudden, these viruses, which have never jumped species before, get just enough information from the environment to be able to then jump. And it's all about adaptation, and it's about learning. And every organism in life, the goal is to live and to perpetuate. Okay. And so whether it's bacteria, whether it's viruses, whether it's worms, whether it's, regardless of what it is, it's to live and breed and continue to populate. And so viruses, in that way, they're extremely smart, and they will figure out ways to create a new host that will then support it. And so that's where some of these outbreaks have occurred, and the transition from animal to human. And viruses can mutate, as we've seen with COVID. It's been an ongoing study in mutations that allow it to continue to evolve and infect and have, even though it's still a respiratory virus, it infects in different ways and it presents in different ways. It's really quite fascinating. So these are two specific viruses within this class of viruses that are especially interesting because of the fact that they have been able to not only jump species, but to cause pandemics. Retroviruses, they are also very interesting because they are RNA viruses. And HIV happens to be a retrovirus. And so its genome is RNA, which means it requires reverse transcriptase to be able to convert that RNA into DNA so that it can then be incorporated into the human host genome. Okay, where then replication and then cellular destruction occurs. So it is an entire process. But what that does, it adds extra steps to the process of viral replication, but it also provides other opportunities for targeted pharmacotherapeutic drugs to be able to stop that replication process. And so when you learn about all the drugs for HIV, realize that look at the different parts in this replication process so that it can be targeted. You have everything from cell entry to various proteases to the reverse transcriptase. And so you're going to see things like reverse transcriptase inhibitors, okay, protease inhibitors. And so these are our classes of drugs that we use to target HIV. Okay, so all it does is add an extra step along the way. Oncogenic viruses, the ones capable of producing cancer. Okay, and so the group that I have listed here include your retroviruses, your herpes virus, adenoviruses, and papoviruses. And so you can take a look at those. These are classified as oncogenic, and I think you can see where the correlation is. SARS-CoV, also known as the infamous COVID-19, CoV-2, because SARS-CoV-1 was SARS back in 2003. It was the original, systemic acute respiratory syndrome, and that was also an avian virus, if I remember correctly, a novel coronavirus. But anyway, this is also an RNA virus, so you can see where this is going to then require that reverse transcriptase as well, which is going to affect its, our targets. It uses protein spikes to bind to very specific cells or very specific surface proteins, AT2, in the alveoli, okay, those that produce surfactant. Okay? And so these are also found in the tubular epithelium of the kidney, in the heart, in the enterocytes, in the pancreas, and in the endothelial cells. So these very specific types, these proteins, cell surface proteins, okay, are the target, which is why COVID-19 has so many negative effects. And people have had MIs. People have had GI presentations with COVID-19, okay? Gastrointestinal, symptoms, nausea, vomiting, diarrhea. And that may be their presenting complaint because it's all about the target, and it has to do with those proteins. So even though it is a typically a respiratory acquired illness, okay, it can have its effect in multiple places, including blood clots, activation of your clotting cascade because of the effect on the endothelial cells. And so this is how its symptoms are so wide and broad and not specific only to respiratory. Any questions on the viruses? No? Okay, we'll move on to bacteria then. And so, as you know about bacteria, and again, a lot of this is going to be review, okay? Again, bacteria don't have an organized nucleus either. They're prokaryotic, okay? They usually only have a single double-stranded circular chromosome of DNA. They may have extra little pieces, which are kind of acquired, which are plasmids, which can acquire, that can code for antibiotic resistance or other types of virulence factors. Okay? But typically, the main genome itself is a single double-stranded circular DNA without a nucleus. Okay. You have the DNA along with RNA and proteins just kind of in the cytoplasm of the cell itself. And it is surrounded by that lipid kind of cellular membrane. But it has a more, your typical bacteria will have a cell wall. Okay. And you know the different shapes of the cell wall. It can be spherical, it can be elongated and box-like, or for your rods, or it can be spiral shaped. Okay. And so, your gram-positives, the cell wall contains peptidoglycan. That's what reacts with your gram-positive stain, your gentian, the violet purple stain. And then your gram-negative actually have an outer membrane of lipopolysaccharide that covers the peptidoglycan so that it doesn't stain that purple. It's going to wash out. It stains, it's kind of a pink. And that lipopolysaccharide is what often confers those endotoxins. So all of those gram-negative rods, especially that have that LPS coating on them, your Proteus species, your E. coli, etc., they have endotoxins associated with that LPS. Once they get destroyed, that LPS and that LPS itself in our body system creates a super antigen effect that stimulates our immune system, and you end up with signs and symptoms of sepsis. Okay. Some of your bacteria have flagella, which allow them to move. So, and there are some of your bacteria have pili and what they call fimbriae, which are kind of protein based spikes or hair-like projections outside of the cell that allow them to adhere to other cells, and often allow them to then gain an anchor to start reproducing and cause infection. Sorry, that's a spam call. So this just kind of goes over your different morphologies, your spherical, just a review. We've talked about biofilms, and there are, just remember, I think of biofilms as there's safety in numbers. So the more you have, the more there are to protect. And so it prevents antibiotics from getting to the central core of the biofilm, so that those bacteria can continue to reproduce and make more, even if the outer ones are getting killed off by the bacteria or inhibited and unable to reproduce. The ones at the core, which are surrounded, they can continue to reproduce. So they have the ability to form these biofilms. You almost never see an isolated bacteria that would be causing infection. It's usually, you've got to have a colony, and then those colonies, again, the ones at the center are always protected. Okay. The replication rates are quite variable. For E. coli, it only takes about 10 minutes for mitosis to occur. However, for Mycobacterium tuberculosis, it takes about 12 hours. So you can see a huge difference in that being a virulence factor and how disease might present. If you have an exposure of 50,000, E. coli, and it only takes 10 minutes for them to reproduce and double their numbers, and then another 10 minutes, you can see the exponential effect and how overwhelming that can be for our immune system. Whereas tuberculosis, it's a much more slow, insidious process. It's not going to present with that overwhelming acute infection unless you have an overwhelming number in the inoculum size. So a large inoculum of MTB can overwhelm our immune system and absolutely lead to an acute, relatively acute infection. Okay. Sometimes when we grow these bugs in the lab, they have very, very classic appearances on an agar plate, or, and odors. Okay. And so, some of the characteristics are used to identify them. Do they have the ability to hemolyze the blood agar plate? Are they slimy looking? Proteus, if you ever look at Proteus on an agar plate, like it spreads out like this, like somebody just did slapped a bunch of paint on a canvas and just let it drip down. That's how kind of Proteus looks on a agar plate. Staph aureus has these big juicy colonies that are kind of creamy with a yellowish hue to them, and they're big and goopy. Whereas like Staph epidermidis, they're very small, much smaller, less robust, less slimy, but they're white. And, you can tell the difference by looking at them on an agar plate. It's pretty fascinating. We've talked about oxygen needs and the difference between aerobes, anaerobes, and facultative anaerobes. And so that plays a role, especially in your pathogens, as you are looking at a patient clinically. Okay. Gram staining versus acid fast. Okay, so your acid fast are going to be the ones the most common is your Mycobacterium species. Nocardia is another one. So if you have a respiratory or even a blood infection or even bone marrow, sometimes you need to do acid fast stains in addition to your typical gram staining culture. And that is a separate order from a, clinical perspective. And realize that, not all species within a genus are pathologic or pathogenic. It's very specific ones that are pathogenic. All right, I am going to give you all a break. Okay, it's about, I will see you back at 12:30. Environment. So, this is where you're really going to find them. Technically, they are all gram negative and anaerobic, but they have pathologic and commensal kind of or saprophytic strains. So in your Leptospira, this has to do with contact with animals who are infected and or their urine. We see this, it's often transmitted by rats, and I believe it's rat urine. Borrelia is the one that you're probably Borrelia and Treponema, the one you're going to be most familiar with Borrelia burgdorferi being the the spirochete that causes Lyme disease. So it has an arthropod vector, the black-legged tick, and treponemes are your syphilis bugs. There is no intermediate vector on these, so these syphilis is transmitted by human to human contact. Okay. So that's really what you need to know, I mean, about these particular, from a pathophysiologic standpoint. Okay. Your mycoplasmas, on the other hand, are very interesting. These are unicellular. They're prokaryotic, so they're not going to have a nucleus and stuff and ribosomes and all that stuff. But they're capable of independent replication, which a virus is not. Okay. However, they're super small. They're like a third of the size of a normal bacterium. The genome is smaller. They don't have a cell wall, so they can have different shapes. Because of the lack of a cell wall, they don't gram stain. And they're actually resistant. Because they lack a cell wall, they're going to be resistant to all of your beta-lactam type drugs that target cell wall synthesis, or any antimicrobial that targets cell wall synthesis, because they don't have a cell wall. And when you think about the mycoplasmas, there are 3 different genera. One is the mycoplasma itself, ureaplasma, and acoloplasma. And both mycoplasmas and ureaplasmas actually require cholesterol from the environment to make their cell membrane. And acoloplasma does not require it from the environment. It does not require it at all. So mycoplasma causes a lot of respiratory, but also causes GU symptoms. There are different species within the mycoplasma. You have Mycoplasma pneumoniae, which is the one you're probably most familiar with, and common cause of community-acquired pneumonia, which is why our standard outpatient treatment of community-acquired pneumonia is not a beta-lactam. It's a macrolide. Ureaplasma, but there's also Mycoplasma hominis, which causes genital infections in males, especially, but it can cause it in females as well. A lot of mucous membrane type of infections. Ureaplasma, again, more in the way of GU infections. Ureaplasma ureolyticum is one of the pathogens. But they won't gram stain. You won't see them on a gram stain, and yet they're pathologic. And so they kind of act like viruses in some way. However, they're still classified as a bacteria, but they're very unique because they do not have a cell wall, and much, much smaller. All right, let's talk about your Rickettsia, your Anaplasma, your Chlamydia, and Coxiella. So these are even more so like a virus because they are obligate intracellular pathogens. They can't reproduce on their own. However, they produce a cell wall, and they reproduce asexually by mitosis, and they have both DNA and RNA, which is very unique because all your other bacteria are DNA containing. So they act more like a combination of virus and bacteria. And so if we look at your rickettsia species or genus, these are going to depend on your host cell for vitamins and nutrients. So again, obligate intracellular pathogen. They do not cause disease in their vectors. They are vector borne. And the most common of these is rickettsia rickettsii, which causes Rocky Mountain spotted fever. Okay. And so transmitted by a tick bite, and it can be fatal. However, with diagnosis and awareness, the fatality percentage has gone down tremendously with this particular disease. Again, Rocky Mountain spotted fever is kind of a misnomer in that, the East Coast here in North Carolina for years, I haven't checked it lately, has had the highest incidence of Rocky Mountain spotted fever. So North Carolina, very far from the Rocky Mountains, but it is something that you don't want to miss because it can be fatal. And we do have treatment for it. Your chlamydia species, these actually will scavenge ATP from the host. So again, it needs the host's ATP intracellularly. It's got to get it intracellularly. It's going to scavenge that. It has a very distinct life cycle. It creates an elementary body, which then transforms inside, and then it will shed elementary bodies again as the infecting, almost like a spore, if you will. It's kind of stripped of everything else. But it goes through this very unique life cycle, which starts as an elementary body, becomes a little bit more developed as a reticulate body. Then there's active replication to make more elementary bodies, which then get shed into the environment to reinfect. And that could be, a local environment within the host itself, or it can be external. It's such a unique bug. And there are 3 species of chlamydia that you need to be aware of, and you'll learn about in ClinMed. Chlamydia, and regardless, they all follow this same pattern. You have chlamydia pneumoniae, which causes pneumonia. You have chlamydia trachomatis, which causes multiple STI type infections, including LGV, urethritis. It causes PI cervicitis and PID in women. It's implicated in Reiter's syndrome. And then there's chlamydia psittaci, that is causes a pneumonia as well, and it's associated with bird handlers. Anaplasma, the Anaplasma ACA, these are very specific. They require the host cell, much like a virus, intracellular. They target specifically host polymorphonuclear cells and monocytes. Okay. There is no defined life cycle like in the chlamydia species. It doesn't require ATP from the host cell. It's independent for energy. And the biggest one that you have to be concerned about and is endemic in this area is Ehrlichia. But the other ones are Anaplasma, Neorickettsia, and Wolbachia. But Ehrlichia, or ehrlichiosis, another vector-borne, infection, tick-borne, mostly ticks. And we have cases of ehrlichiosis here in Maryland, and where there's, I forget, I think it's transmitted, this is actually transmitted by the dog tick, if I'm not mistaken. And so the what you will see is, because it targets the host polymorphic nuclear cells and monocytes, you can see somebody who has a neutropenia and a monocytopenia. Okay, so if you look at their blood smears and stuff, typically we're going to do, immunoglobulin testing, antibody testing for this. And it's not something that you're going to really grow because of the fact that you're only going to, it's intracellular, and it will lyse the host cell when it releases its, I guess, progeny. Coxiella, there's only one. It's Coxiella burnettii, and I think the D is an accident. And this has multiple human hosts, multiple animal hosts, but it causes specifically Q fever and Q fever in humans. Again, these are bacteria that are kind of a blend between bacteria and viruses. So we're going to move on to fungi if there are no questions. Okay. Your fungi, they are free living, eukaryotic. They do have nuclei. And, it's not all of them that are capable of disease in human. There are a few. Most of the time, as we've talked in AMS, these are going to be superficial skin type infections, and except if you have a loss of immunity, like if you become immunocompromised in any way, or even immunosenescent, as in the aging immune system. So it can be fatal in your immunocompromised. These infections can be fatal in your immunocompromised hosts. Your fungi are divided into yeasts and molds. Yeasts are single-celled. They reproduce by budding. Some will form pseudohyphae, like your Candida can form pseudohyphae. But your molds, they grow by hyphae. Like, so it's your typical bread mold that has that fuzzy appearance and kind of like this meshy, fluffy mesh network that, and they make spores, and then the spores get released, and they will, wherever they land, they can create new molds. Okay. Some fungi are dimorphic in that they can change from a yeast to a mold depending on temperature. So they can, at one temperature, they might appear like a yeast, and then you heat it up, and then they're going to look like a mold. So, and your three main species there are blastomycosis, histoplasmosis, and coccidioidomycosis. And you'll learn about these in ClinMed as well. Let's see. But, normally with an intact immune system, and again, this is mostly your T cell immunity, and because we have normal flora that competes for nutrients, the fungi are normally kept in check. It's only when there are disruptions in that balance that we end up with, overgrowth or, infection with some of these organisms. All right, parasites. These are grouped into protozoa, helminths, and arthropods. These are actually members of the animal kingdom. And so your protozoa are unicellular, and they have, full cellular machinery and can function outside of the host cell. They're not going to have cell walls because they're animal kingdom, and so they will have cell membranes. Your biggest protozoans, your more common protozoans are malaria, amoebic dysentery, and giardia, causing giardiasis, and so those are relatively common. Your helminths are worms. They're subdivided into 3 different types. You have your roundworms or nematodes. You have your cestodes, which are like your flatworms, tapeworms. And then you have your trematodes, which are your flukes, like schistosomiasis. Okay. And so these are all, again, and yes, people do get worms. They still get worms. You know, they're not the only, it's not the limit. There are other ones you'll learn about, pinworms and hookworms and stuff like that. And then, of course, we mentioned arthropods, which often are vectors for disease. And so it includes vectors as well as some of our ectoparasites. So, and when you think about ectoparasites, you're thinking about lice, chiggers, scabies, all those pleasant little critters. All right, so those are your basic categories of disease-causing organisms. And, you know, and you can see that there are clearly some differences, you know, amongst them. And those that have, let me just go back a second. So when we start talking about, Dr. Noel starts talking about some of our anti-helminth drugs, and, you know, you're going to see, because they contain all the machinery, we can target very specific, you know, mitochondria and different things within these cells, within these organisms, to actually destroy them. But what won't work are things like cell wall inhibitors, etc. So these characteristics become really important when you start thinking about treatment. Okay. And so keep these in mind, because that's going to help you figure out why certain antimicrobials work and others don't. So, all right, let's talk about mechanisms of infection. Okay. And, you know, I mentioned it in the beginning when we were looking at the triangle, the epidemiology of infectious, you know, epidemiology side of things. That's kind of like the environmental side of things and how that impacts and, you know, all that good stuff. This is really about epidemiology, as you've learned, studies patterns and determinants of health, hoping to control disease and other health problems. So it's, you know, all these social determinants become part of epidemiology. If we focus, okay, there's both descriptive and analytical epidemiology. So your descriptive is going to look at these gross patterns, but the analytic part of it is going to look at the different factors influencing the disease. Okay. And then, you know, if you target factors within the infection triad plus the modes of transmission, so you're going to target those in order to control disease, etc. So you hit it from a couple different aspects when you're looking at it from an epidemiologic standpoint. Okay. Who wants to tell me what the incidence of disease is? What that means? Incidence?
I believe incidence of the new cases.
Very good. In a given environment, okay, within whatever block of time, including typically a year. That is your incidence, okay? It's the number of new cases, okay? How about prevalence?
prevalence, the total number, like the new plus the current or something like that.
Correct. It's
I
those living with disease. Okay. It's those living with disease. Okay. All right. What's the difference between endemic, epidemic, and pandemic? You can take any one of them if you want.
Pandemic is global.
Okay, so pandemic is global. It's outside of a select geographic area. It's just like when we pan culture somebody. It's whole hog, okay? We're gonna culture everything. Pan is, it is. It's more global. It's spread everywhere. Okay, good. What's epidemic?
Isn't that when it rises above the normal baseline level within a population?
Correct. So it's when it rises above a normal level within a given population. And, so Jalen, you can say community, but it's within a given population. So you can have, epidemics within a country or within a city, or like right now, we talk about the opioid epidemic, okay? And it's more than a community. It's really within, we're talking specifically within the United States. Okay. And so it's within a population, whatever that population might be. And Aaron, you're correct. Endemic is what's something that occurs regularly, but is very specific to a particular population or geographic area. Okay. And so, and we talk about this a lot in travel medicine, because if you're going to go to a different country, it's best to go to a travel clinic, or look it up yourself, find out what diseases are endemic to that population or that region, geographic region. Is it malaria? Is it hepatitis A? Is it typhoid fever? And you've got to look at those things and perhaps get vaccinated or given immunoglobulin prior to going, to help boost your immune system, since you've never been exposed to it and not previously vaccinated. Okay, so but those living in these environments, this is normal to them. Okay. All right, let's talk about modes of transmission. Okay. And I'm going to, I will say that most pathogens, will present based the way they transmit is often, their portal of entry. And think about all your respiratory viruses. Okay. But it's only most. It's not 100% rule. It's not an always. Okay. Because we have other pathogens that might present or get, the way they're contracted is through the GI tract, but they find their way into the bloodstream, and then they make their way to the respiratory tract, and then they get transmitted that way, or they get filtered by the kidneys and urinated out. So then it goes out in the urine, the GU tract. So, and schistosomiasis, one of your flukes, is very fascinating in that it can burrow through intact skin That's its portal of entry. It doesn't even need, a breach in integrity of the skin. The worm can burrow through the intact skin, make its way into the bloodstream, end up in the liver, end up in the GI tract, where then it produces its cysts and trophozoites and that get excreted in the feces for it to then resume its life cycle outside of the human. We are just one step in the process. So the portal of entry doesn't always have to be the same as the mode of transmission. So it is kind of important that from an infection control standpoint, that you study your bugs and know some of the unique pathogens. Penetration is also important in transmission, and penetration from, can be anything from, an IV stick, a needle stick, a puncture wound, a broken bone, a compound fracture that reaches the integrity of the skin. It can be anything along those lines. It can be from a catheter that is inserted through the urethra into the bladder. Okay, so all of those items are kind of iatrogenic way. Most of them are iatrogenic ways of, of allowing for penetration. However, bacteria themselves sometimes have those pili and fimbriae and or other virulence factors that allow them to gain penetrance into the skin and deeper tissue layers. Okay. There are modes of transmission via direct contact. Okay, and most of our ectoparasites are transmitted that way. However, some of our STIs, including syphilis, and syphilis, Haemophilus ducreyi, which causes chancroid, and even chlamydia, it's direct contact. And so that allows these bugs, and I use that term lightly, to transmit from one host to another. Okay. Another mode of transmission has to do with ingestion. Okay. And so when we talk about ingestion, it's usually fecal oral is a specific mode, when you consume something that has been contaminated. However, it doesn't always have to be fecal oral. Fecal oral, it can be specifically contaminated product, like this latest listeria outbreak. And so Listeria monocytogenes is a vicious gram-positive rod that causes significant maternal fetal morbidity and mortality. And so there's a huge outbreak right now that's been ongoing since the beginning of January in, again, soft cheeses, and it's pretty significant. Okay. But when we ingest foods, think about the pathway, and we start out aerobic, we end up anaerobic, we have stomach acids. So, we have protective mechanisms. And so these pathogens have to be able to withstand some of those extremes of acidity, those acidophiles. And so, yeah. And when we ingest, is it the pathogen? Is it a spore, like in your Clostridial species, or is it a toxin? that's already an exotoxin that has been produced, and you're ingesting the toxin, which then causes disease. Right? So these are some of the things when you consider the mode of transmission via ingestion. And then, of course, inhalation. And is it the pathogen itself, or again, is it spores? There aren't as many spore forming respiratory infections because a lot of your spore formers are anaerobic. Okay, if you look at all your clostridial species. And however, anthrax is something that can be inhaled and cause pulmonary disease via spores. So always consider your mode of transmission. And, you know, this is from a, and when we talk about like direct contact, okay, I'm going to jump back to that for a minute because I just had a thought. It doesn't always have to be direct person to person. And so when we think about daycare centers and even some long-term care centers and stuff like that, there are plenty of fomites, healthcare acquired infections. There are plenty of fomites that are capable of transmitting. It's still a direct contact kind of transmission. However, it's on some other inanimate object that has been contacted. And we know that certain bacteria, certain microbes, they can only last so long in the environment. However, other ones can last a lot longer. And spores pretty much last the longest. They can exist for years and years in a dormant spore-like phase that then can, given the right opportunity, cause disease. Okay. Your source of infection, okay, and again, this all goes back to what kind of questions you're going to ask in your history and physical. You know, the source, this can be where, is it a location? Is it a particular person? Is it an object? Was it a substance that contained it? You know, and source of infection, it can be quite variable. And again, these can be endogenous or exogenous. Human, inanimate, animal vector, arthropod vector, just look at all the things we've already talked about. All of these can be sources of infection. It can be a location, whether it's like hospital-based or healthcare associated or community. It can be geographic. And then it can be a specific body fluid component. So it could be feces or urine or bloodborne infections, etc., or your STIs when it comes to semen. Right. So when we look at how diseases manifest themselves clinically, okay, and the course of a disease, okay, we're really talking about the signs and symptoms over the duration of the illness. And so there are very distinguished stages of disease, and that is going to be variable amongst the different pathogens and individuals. So, you know, I mean, when I got COVID, you know, my gut told me it was going to happen. And, you know, I knew I was like, this is a bad environment. I should not do this. But I did it anyway. And it wasn't even 2 days after the exposure. It wasn't even 2 days that I came down with symptoms, you know, and other people, it might have been 10 days or a week. And so it's quite variable. Again, depending on all of these different things we're talking about, inoculum size, virulence, strain of the virus, you know, immune status, etc. So, you know, but your 5 distinguished stages of disease include the incubation period, and that's, after you've been exposed and the pathogen starts to reproduce, but not causing any symptoms, nothing recognizable. So it's an incubation period. So you've got a certain inoculum size. You've got it until your body starts to react to this particular pathogen, and it builds up its numbers to actually cause an effect. Then you may not have any symptoms. Your prodromal phase is when you start to have appearance of symptoms, but a lot of times these are vague during the prodromal stage. They're just kind of more generalized and not specific to the infection itself. It's just generalized effects of your immune system. And then, of course, you have your acute stage, where that's where you're going to experience the maximum impact of whatever infection it is. And you are going to, and at that point, your pathogen count's high, the replication rate is high, the transmissibility is high, and that's your acute phase. You have your convalescent stage, and that's where your immune system has contained the infection. It's not, it's starting to eliminate it, but it's not completely gone. And with all of the release of all the cytokines, you've got a lot of tissue damage. And so your body's starting to repair that. However, it's not back to normal yet. And then you have your resolution stage. And so your resolution is when the pathogen's completely eliminated, and all of the damage has been kind of repaired or corrected so that there aren't any residual signs. And that means your complete resolution. So those are your 5 stages. And they can vary among the different pathogens and among individuals. Some are going to have a very specific 3 to 5 days prodrome, or 2 weeks of incubation, followed by 5 to 7 days of a prodromal, and it's variable. And so when you come across these, and you're going to talk about these in peds if you haven't already, a lot of your childhood illnesses, they have very specific patterns, and the patterns alone are what allow you to clinically diagnose what the pathogen is. Especially with your viral xanthoms, chicken pox versus measles versus, roseola versus rubella, all of these different things, that they have very specific patterns to them and prodromal phases. So incubation, prodrome, quite variable depending on the pathogen. Your peak here is going to be where your symptoms are at their worst. Oftentimes we will see, as during this upstroke of worsening of symptoms, is that's where death is going to occur in certain individuals, when they surpass that critical threshold, and just overwhelms the body. There are people who will have subclinical disease and may never experience symptoms, and we've seen that with COVID as well. And people who are asymptomatic, and yet they're spreading the virus as well, because it's actively reproducing, and they're spreading disease, but they don't know, because they haven't reached the clinical threshold to say, I'm symptomatic, I'm having symptoms, because it never reaches that level. And then as opposed to those who go through convalescence and resolution, there are specific patients who have chronic disease, and long COVID is a great example of that. And so they are chronic, have chronic symptoms that they're not resolving. And so COVID has really been quite an education in all of these kind of stages of and variations within this infectious, the stages of infection. We talk about the sites. It's you just add itis, to whatever the site is. If it's rhinitis, if it's sinusitis, if it's pharyngitis, if it's bronchitis, you just add itis to it. Okay. It doesn't really distinguish between infectious and non-infectious. It just tells you that there's some kind of inflammation there. And so that's why bronchitis, it's generic. Sinusitis, it can be viral or bacterial. They're generic. It just either one can cause inflammation. You treat them very differently. So emia means that it's in the blood, whatever it is. So if you have bacteremia, it's bacteria in the blood. If you have viremia, it's viruses in the blood. And sepsis is, and we'll go more into sepsis and SIRS next week, but sepsis is essentially a very dysregulated post-response infection, and that leads to multi-organ dysfunction that can be potentially life-threatening. Right? And this is right now an ever-evolving definition compared to what the traditional is. So traditionally, there were criteria that had to be met to say somebody was septic. They had to have a certain fever or hypothermia. They had to have a certain heart rate or and respiratory rate and a certain number of white blood cells. Not anymore. Okay? So that's kind of the old traditional definition, which has been morphing over the last several years. And when we talk about sites of infection, is it localized or is it disseminated? Okay? That is an important distinction to make. So when we look at localized infections, like pharyngitis, okay, can you still have fever, body aches, all of that? Yeah, but it's pretty much localized. Disseminated is when, it's probably been transmitted or there's been some hematologic spread or access to the systemic circulation. Okay? And those are the patients that are going to typically crash and burn much quicker, those who have disseminated disease, okay, as opposed to localized. All right, virulence factors. So we have, what time do I have to? I have till 1:45. So I guess I gotta crank it up a little bit. Virulence factors, we've talked about toxins. They're really super antigens, okay? And I'm gonna go over that. Exotoxins, they're released from the cell. Your endotoxins are that LPS that are in the cell wall of your gram-negative, okay? And so, these are inherent to the bacteria themselves. They don't have to make anything, manufacture anything, release anything. Exotoxins, if you have a toxin-producing organism, then you've always got to be wary if you kill off that particular bacteria and you destroy that, it's going to release those exotoxins. And sometimes that massive release of exotoxin is enough to cause a fulminant response in the body and even death. It can make the patient sicker as opposed to monitoring. So some of our GI infections, even with pathogenic bacteria in a normal, healthy individual who is immunocompetent. Sometimes we just watch and wait instead of trying to intervene with antibiotics because of the risk of killing off all these bacteria and massive release of exotoxin, which then can shut the kidneys down. So when you balance these, this is part of the pros and cons in your decision-making process when it comes to treating various infections. We've talked about adhesion factors. They can be very site-specific, so a certain predilection for certain tissue types, cell-specific, non-specific. And so there's a lot of different adhesion factors that are out there that add virulence to organisms. Okay, evasive factors, I have a whole slide on that. We'll get to that. And then there are invasion factors. So some bacteria are able to produce, mucinase or proteases or elastases, collagenases, keratinases. And if they're able to produce it, they can break down and utilize, that environment for the nutrients. And, so we talked in AMS about, your fungal, your dermatophytes, and why they produce keratinases, which then can break down those keratinocytes, release any of those proteins and nutrients, and utilize that for their growth and reproduction. So having these certain abilities can sometimes allow the organism's access. Some virulence factors, this is a summary of certain microorganisms and with their virulence factors. So, Vibrio cholerae, which is a specific organism, bacteria, produces an exotoxin called cholera toxin, and it causes a terrible secretory diarrhea. Okay. And so the body will try to dilute it, wash it out. And so it's a secretory kind of diarrhea, non-inflammatory. So you're not going to have the blood and you're not going to have the mucus and stuff like that, but lots of watery diarrhea. And then you can go down through some of these and, and you can see what some of these organisms can do. And it's just kind of a summary, takes a look at specific pathogens. Okay, so toxins as superantigens. What this means is essentially it's going to trigger, it's an antigen, it's going to cause an antigen antibody type reaction, it's going to trigger the immune system. Okay, it can react with the human leukocyte antigens on your antigen presenting cells. That's what HLA on APC stands for, human leukocyte antigen on your antigen presenting cells and your T cells. And so then it can induce both local and systemic responses. So these toxins essentially become these super antigens, which then can cause either significant local, as in the case of some of your secretory diarrheas, or it can cause systemic reactions like botulinum toxin. So your diphtheria toxin can inhibit cellular protein synthesis. That's one of the nasty things about diphtheria, one of the reasons we vaccinate. Botulism, it's going to decrease your release of acetylcholine, and it's going to lead to flaccid paralysis. You're not going to be able to contract your muscles. Tetanus toxin, it's going to release neurotransmitter from inhibitory neurons, and this is going to cause a spastic paralysis. So one causes flaccid paralysis, one causes spastic paralysis, and then cholera, which is the diarrhea. Okay, there are 5 groups of these super antigens. And 1 through 5, 1 and 2 really cause serious disease in human. And so if you look at strep pyogenes, it can produce up to, and you've heard about strep pyogenes. Yeah, it causes, strep throat. It can cause rheumatic fever. It can cause scarlet fever. Well, it produces up to 11 different super antigens. And so it can cause a whole host of problems. Glomerulonephritis, autoimmune disease, myositis, toxic shock. So, it has to do with the specific strain of the strep pyogenes and kind of which of these super antigens is it able to produce, which direction it goes as far as disease processes. Okay, why does one person get acute rheumatic fever and one person ends up with scarlet fever and one person ends up with post streptococcal glomerulonephritis? This is why. Okay, it's these super antigens. And remember, the glomerulonephritis, rheumatic fever, they're immune reactions. They're immune reactions. It's to these specific antigens. Staph aureus can produce up to 24 different super antigens. You thought 11 was bad? This is why Staph aureus is so virulent. And yeah, it can cause all kinds of a whole spectrum of disease. So exfoliative toxins, and you can have the food poisoning, you can have toxic shock. Okay, so you can have the staph scalded skin syndrome. So there's a whole host. Okay, and yeah, there are other ones as well that can produce super antigens. All right, looking at the evasive factors. Okay, these pathogens, we talked about, some of them have this slimy coat. So capsules, slime, mucus layer, whatever you want to call them. And that's going to kind of hide their surface antigens. It's going to allow them to, propagate within the host without being detected. It can inhibit phagocytosis. And so, we look at things like strep there, especially your strep pneumoniae, Neisseria meningitidis, your Haemophilus influenza B. And all of these are, have significant effects on neonatal mortality. Leukocidin C, again, leukocidins produced by Staph aureus, they can destroy neutrophils and macrophages. So our own immune system may recognize it, but if they release this, these leukocidin, it can actually kill these white blood cells off, which are our first responders. Salmonella, Listeria, Legionnaires, they actually reproduce after they've been engulfed by polymorphic nuclear cells. Okay, very interesting. H. pylori, you remember, I hope, about secreting urease, which converts the gastric urea to ammonia, which neutralizes the acid and allows it to live in that acid environment. Staph aureus a surface protein A can immobilize IgG. It can also produce coagulase. H-flu GC inactivates IgA in the respiratory and GU tracts. So we know that IgA is in our mucous membranes, and Haemophilus influenza and the gonococcus can inactivate that, has the ability to inactivate our own IgA in those mucous membranes in the respiratory and GU tracts, which allows them to evade some of our normal defenses. And Borrelia can actually alter its surface antigens to avoid detection. Fascinating. So fascinating. So how do you diagnose and treat these? Okay. I'm going to cruise through this pretty quickly because this is kind of getting into the clinical medicine side of things. But again, you always start with your H&P. Okay? And so what's your patient's symptoms? What kind of signs? Always look at your vital signs. That can give you a clue as to whether something's systemic or local. And again, that those vital signs are going to kind of really guide your decision making as to how quickly you're going to act, react, intervene, etc. Okay, and yeah, you have to have knowledge and or look up if there are things like what season is it? Are there any seasonal outbreaks? Are we in flu season? Are we in parvovirus season? And are there local food related outbreaks? That can become, especially with your GI illnesses. We often will group your physical exam into organ systems, your review of systems, your physical exam, realize that your heart and lung is always paired. So if you're going to ask a heart question, you better ask a lung question. If you're going to ask a lung question, you better ask a heart question. With HENT, you got to be specific to infectious etiology. If you're thinking infectious, I don't want to hear about, glaucoma and, stuff like that. I don't care about dentures. I don't care about when their last dental appointment was, unless it's very specific to a tooth abscess. So tailor, what you're saying or asking or, viewing. It is important to ask about recent travel. And is that travel local or overseas? Because that's going to change your perspective as to what are your potential pathogens. Okay. Did they travel to a disease endemic area? And then you always have to ask about recent exposures, and that could be household. Is anybody in the household? You have any children that are in daycare? I mean, it's as simple as that. But it could be something like, oh yeah, well, I've been going to visit my mom in the nursing home. So at that point, you're exposed to, potential hospital acquired infections. So recent exposures is, it can be quite broad. And then you always have to ask about patient-specific risk factors and looking for, chronic illnesses like diabetes, hepatitis, liver cirrhosis, renal insufficiency, that, or autoimmune diseases. Are they on any, immunosuppressive drugs? Very specific risk factors. Okay. Your diagnostic studies start with your cultures and sensitivities. Obviously, you've got to consider your serologies. There's always PCR techniques, your different RNA, DNA sequencing, your rapid tests for things like flu and COVID and mono. And then, there's all kinds of others that you learned about last semester. Okay. And so, this chart is in your text as well. And I put it in here, not that I'm going to ask these questions myself, but it is a good reference for your antibiotics, okay, your antibacterial agents and how they work. Some of their examples, target sites, and what are the side effects. We also have antivirals, which Dr. Noel will talk about, and most of these are synthetic. And again, many will target viral replication, but some will target the attachment to the cells. Are antiparasitic, really exploiting metabolic processes within the parasites, and then trying to stay away from stuff that's shared by the host, so you don't have negative effects in the host as well as killing the parasite. It's going to be parasite specific. And then antifungals, which will most of them interfere with the ergosterol in the cell membrane of the antifungals, or inhibit the synthesis of the ergosterol. But these are how we attack those particular organisms. There are some other interventions, including immunotherapy. And so, we talk about immunoglobulin, intravenous immunoglobulin, where we can actually give patients immunoglobulin that's been prepared, that already has antibodies from other human donors, okay, and that's infused into the patient. And so we've used that with COVID, so monoclonal antibodies. We have intravenous, IVIG. These are really reserved for your sick of the sick patients. We have some genetically engineered cytokines, including interferon and interleukins, that can be infused in some cases. But your primary intervention are vaccines. There are sometimes surgical and interventions that have to be done in order to debride areas and remove areas of infection. Sometimes you have to remove prostheses because of biofilms. And you always have to look at your long-term outcomes. And then I'm just going to talk about, I think I have one slide on bioterrorism and global infectious diseases. So you live in a world that, and this is going to be ever evolving, where bioterrorism is a valid threat. Okay. And so, it wasn't until 9/11 and the anthrax scares, and actually some of the anthrax cases, that we really ramped up and prepare for bioterrorism. Okay. And so there is a whole laboratory response network, which, in order to try to identify any bioterrorism, so that there can be a very rapid, coordinated detection and identification. And, many people have thought that COVID was designed for bioterrorism. And, we don't have any evidence to either completely deny it or to support it at this point in time. But you have to realize that the threat is real. Okay. And so there's a whole list of, and categorization of bioterrorism type of pathogens. And it ranges from things like anthrax to down to like even STIs and stuff. But, and with respect to probability and all of that. But there's a whole branch of the CDC that, focuses on the bioterrorism. And I think that, it's something that you have to be aware of. And what they're tapping into is all of the things that we just talked about. So, the transmissibility, the ability of whatever it is to be in the environment. So spores are very good. Can you aerosolize these spores and and infect a large population? And so that's what they're looking at. And obviously the best bioterrorism um, kind of weaponry is going to be something that can infect a large population and be easily transmissible or transportable and aerosolized and be in the environment to have its effect. So it's just something to be aware of. And we're always monitoring for some of these emerging global infectious diseases. Many of these now are zoonotic, in origin, meaning that they come from other animals or vectors. And part of this has to do with, just man's, continual expansion into previously uninhabited areas. And that's going to happen when you start having man and humans and animals and other critters coming closer and closer and closer contact. There's going to be more and more transmissibility. Okay. And so some of the more, worrisome emerging infectious diseases, malaria is always a potential, especially in the United States. And there's a lot of theory with global warming that, we're going to start seeing malaria in this country because of mosquito-borne vectors. We're seeing West Nile virus. There have been outbreaks, and there is no real cure for it. It's just something that we can support. And somebody's got to mute. Somebody's got their and then of course we've learned about SARS-CoV-1 and 2. So there's Zika virus, which there was a big outbreak several years ago. And that's a concern as well. Another mosquito-borne virus. And then Ebola, which we had an outbreak, I guess it's probably been about 10 years ago now, maybe not that long, but maybe about 10 years ago, there was a big Ebola outbreak. And, but, Ebola is something that is endemic to certain countries in Africa, in West Africa, especially. And so, again, these, if they get more broadly disseminated, it can wreak havoc in our healthcare systems. I don't know if anybody remembers the Ebola outbreak, and there was a guy from Liberia, I believe it was, who died, but it wasn't, he had already infected a couple people in a Texas hospital. And they misdiagnosed him and sent him home, and he ended up dying. And then other people got really sick, and then they were quarantining people right and left. Anyway, that's pretty much all I have for infectious diseases for today. Any questions?